My name is Alaina Nichols.
And I'm Mattison Mclain.
And the enzyme that we chose to study for this project is luciferase. Luciferase is responsible for the bioluminescent reaction between luciferin, oxygen, and luciferase. And bioluminescence can be seen in this picture. And luciferase is an oxidative enzyme, which is why it reacts with luciferin and oxygen in order to ultimately form a ketone onto luciferin. And it is in the beta-clam family, meaning that it primarily consists of beta barrels in each of its 3 homologous domains. It is in the lipocalins class, and lipocalins basically means that it binds a hydrophobic molecule, such as luciferin in this example.
So we chose a particular dinoflagellate to study, which is called Lingulidinium polyedrum. And this particular dinoflagellate has luciferase in it, which is what we're studying. And so we're going to kind of take a look at the dinoflagellate first to see what's going on in there. So this is a dinoflagellate model. And the one place we really want to focus on is the vacuole inside of the cells here. So the vacuole is highly acidic. And this is really, really important in the reaction with luciferase. So what happens is that we have these dinoflagellates, and they're just kind of floating in the water. And anytime that there's a stress that's caused on them, so it might just be somebody moving their hand in the water, it could be a wave, it could be anything like that at all. Whenever there's a stress, it sends an action potential across the vacuole. And what this does is it causes the vacuole to release the protons in there that are causing it to be highly acidic. And when the protons escape, it makes the entire cytoplasm inside the cells very, very acidic. Now, inside these particular dinoflagellates, they have these things called scintillins. And scintillins are subcellular particles, and they just kind of float around. And these scintillins are unique to these dinoflagellates, so they're unique to the polyedrum dinoflagellates. Now, if we go back and we think about it, so we have this action potential that goes across it. We have the protons that come out of the vacuole and they acidify inside the cytoplasm. Well, what it does is it causes those scintillins to become acidic. And what happens is when they become acidic, they emit flashes of blue light. And that's how we get the bioluminescence. So, pretty interesting. It's very unique. So this here is the isolated scintillins inside the cytoplasm. And you can see here, they're super tiny. And they emit this blue light. So pretty cool. Yeah. They hypothesized that this is regulated by proton-gated channels, but they're still not sure. There's still a lot that's unknown about luciferase. But we've done our best to figure out everything that we can. And yeah, so very cool stuff.
The structure of luciferase is really, really important to how it's regulated and its role with binding luciferin. Luciferase basically just acts like a cup and put your hand over it to regulate it. So the beta barrel is made of antiparallel sheets, and there's 10 of them. It's beta sheets 5 through 14. And the 3 helices that cooperate as the lid are alpha helices 2, 5, and 6. Alpha helice 2 is more inside of the beta barrel when it closes, so it kind of acts like a latch. And it latches onto beta strands 3 and 4. So it binds hydrophobic molecules such as luciferin at low pHs based off the proton-gated channels. So the active site for luciferase is referred to as the putative ligand binding site, and it can be seen right here. In this example, there are a few molecules that have been stripped away just so it's easier to see the active site. But it is also bound to water molecules because luciferase is bound to water molecules when it is at a basic pH, such as pH of 8. But when it is at a pH of 6.3, which is its optimum pH, it will bind to luciferin instead, specifically by using histidine 924, which is bound to a serine 921 through hydrogen bonds, and also a glutamate. And this creates a hydrophobic binding pocket, and that's where the luciferin will sit during their reaction. Another important histidine is histidine 899. This one is the one that helps this the helices hinge over top of the beta barrel. And the hinge is made up of a bunch of glycine residues, as well as being regulated by this histidine residue. And histidine 909 and 930 are just other pH regulating ones, but they don't play as important of a role as histidine 924 and 899.
Awesome. So we had to dig through a lot of journal articles and a lot of research to figure out anything about luciferase, because not much is known, and all of the research that has been done is not readily found. So it took us a while, but we found a lot of articles, and the one article that we focused on was an article that studied the biological clock of the dinoflagellates and how that controls luciferase activity. So what they did is they took dinoflagellates and they started growing cultures of them in a lab. So they took the cells and they wanted to see what would happen if they just observed them just over a couple of days to see what would happen to them. And then the second part of the experiment was that they wanted to add antibodies to the cells to figure out if it would inhibit the reactions that were going on or what would happen. So they created anti-luciferase antibodies that they created inside rabbits, and they put those inside the cell cultures to see what would happen. And the results were that they figured out the mechanism. They figured out what happens, like what's going on with luciferase and how the entire mechanism works. So I'm going to go into the mechanism because that's what the journal article explained. So this is a luciferin molecule. This is the protein that's involved. So if we think back, so we have all of these scintillins, which are the particles that are floating around in the cytoplasm. And you also have these proteins that are they're floating around in there. And what happens is that we have the action potential that goes across the vacuole. We have the protons that come out of the vacuole. And then what happens is when we lower the pH, so it's normally at a pH of 8, when you have more protons coming into the cytoplasm, it lowers it to about 7. And when it hits that 7 point, it starts to create a conformational change in the protein. And the reason why it has a conformational change is because we have another protein called luciferin binding protein, and that binds right here where this purple arrow is. So when it's bound, it kind of keeps it cupped over like this. Well, when we bring it down to 7, the luciferin binding protein pops off, and then it opens up. And when it opens up, it leaves this area open for a reaction to occur. So when that opens, luciferase also becomes active, right around that pH of 7. So luciferase comes in, and it comes in with the oxygen, so it comes in with some O2, and it hits that area right there, and it pops on a ketone. And it's amazing because just adding that ketone onto it causes those big blue flashes of light. And it causes blue light to flash at around 480 nanometers. And that's the whole mechanism. It's actually pretty simple. So I'll show you another picture of the conformational change. So this one right here, this is after it's opened up. So the luciferin binding protein has already left. And we got this area here open. So luciferase comes in with oxygen, pops on that ketone, and then we create light. We create this oxyluciferin, so it becomes oxy because we've added the ketone onto it. Oxyluciferin, and then we also have a product of water. Pretty cool. This is the same thing here. It just shows you that there's a pH of 8, and when you lower the pH, you get all these wonderful products, and we have bioluminescence. So yeah, pretty simple.
Some related proteins to luciferase are the 6 other forms of luciferase found in various organisms, as well as human fatty acid binding protein and retinal binding protein. Retinal or human fatty acid binding protein and luciferase are 18% identical and 38% similar. And the fascinating thing between all 9 of these different proteins and enzymes is an arginine located at 1142 of every single one of them. This allows all of them to bind a hydrophobic molecule due to the release of water or the breaking off of hydrogen bonds. And it's just very interesting that all of them work because of one amino acid residue in every single one of them.
Yep.
And it's important because bioluminescence is trying to be studied in order to develop some form of tracer protein, or so that can be attached to certain amino acid residues so that it's easier to sequence proteins, just being able to see things, and for proteins that we can study exactly how they work, just based off bioluminescence being present.
And then the final thing that we also learned is that it is controlled by the circadian biological clock. So that article, that primary article, told us they figured out that if they observed the cell culture, so all those dinoflagellates, that over a period of a day, the scintillans, so at night, they were active at night, and then they would destroy themselves. And then it would just keep going on, after 24 hours. So that was the other thing, that was the final thing they left us with. So they are pretty unique, and Luciferase is pretty cool. So we're gonna leave you with a really awesome video of what's going on, and it shows you that circadian biological clock that's going on. Shows you how it's only active at night, and if you were to do this during the daytime, you wouldn't see anything. So we'll show you the mechanism and what's actually going on.
But first, if you have any questions, just comment below, and we will get to them as soon as we can. But here's the video.
So, pretty cool. They just dropped a rock in the water. Then you can see all those dinoflagellates glowing. So, pretty awesome. So, thank you so much for your time. And our references. So many references.
Yep.
Thanks so much.